Hello friends, myself Professor B.D. Bhoknal, Associate Professor, Botany Department, KTHM College, now Nasik. Today's topic is now lysosomes and ribosome. It is from cell organelles. From the subject now the cell biology and molecular biology. So today's topic is cell organelles, and within that now the study of lysosome and ribosomes. Lysosomes, these are found in cytoplasm of all types of eukaryotic cells. These were first discovered by D. Dewey in 1955. These were predominantly present in the animal cells. It is secretory in function. Example: pancreatic cells, hepatic cells, leukocytes, spleen cell. These cells contain large number of lysosomes. Morphology of lysosome: the size and shape in change according to the type of cell. They may be spherical, oval, or biconvex in shape. They may be 0.2 to now zero point 0.8 mu in diameter. In the cells of mammalian kidney, these are of big size up to 5 mu. Lysosomes are bound by single unit now membrane. Types of lysosome. There are four types of lysosomes. These are follows as follows. Primary lysosome. These are also known as pre-lysosome. They may be derived from endoplasmic reticulum or Golgi body. These are small sac-like structure and it contain enzyme. Secondary lysosome. These are also known as digestive vacuole or now heterophagosomes. These are formed by combination of pre-lysosomes with pinosomes or phagosomes. Third, residual lysosome. These are formed due to the lack of certain enzymes in secondary lysosome. This results into the partial digestion of food material. Therefore, these lysosomes contain residue, hence the name is given residual lysosomes. Example: hepatitis, it means the inflammation of liver, then the joint injury. Autophagic lysosome. These are also known as autophagosomes or cytolysosome. These are formed when cell feed or its intracellular organelles such as mitochondria, ribosomes by autophagy. It has been observed that during the starvation of organisms which feed on cellular component, sometimes lack of oxygen in the cell also brings rupture of lysosome. They release their enzyme in cytoplasm. Cell death takes place due to autodigestion. Hence, these are also known as suicide bags of now the cells. Structure of lysosome. Structure of lysosome. Lysosomes are without any characteristic shape or structure. Example: they are now pleomorphic. They are mostly globular or granular in appearance. It is now zero point 0.2 to zero point 0.5 millimicron in size and is surrounded by single lipoprotein membrane, unique in composition. The membrane contains highly now glycolated lysosomal associated membrane protein, that is LAMP, lysosomal associated membrane proteins. LAMP, and lysosomal integral membrane protein, that is LIMP. LAMPs and LIMPs form a coat on the inner surface of the membrane. They protect the membrane from attack by numerous hydrolytic enzymes retained inside. The ribosomal membrane has hydrogen proton pump, which is responsible for maintaining the pH condition of the enzyme. The acidic medium maintained by the proton pump that pumps now hydrogen inside the lumen ensure the functionality of the lysosomal enzyme. Inside the membrane, the organelle contain enzyme in the crystalline forms. Functions of lysosome: intracellular digestion. During starvation, lysosome digests the stored food material. Extracellular digestion: lysosome brings about digestion of food particles which are suspended at the intercellular space by discharging enzyme outside the cell. Autolysis: lysosome digests the various organelles of cell, example mitochondria and ribosomes. Metamorphosis: in amphibian and insect, lysosomes are responsible for metamorphosis. For example, tail of tadpole in frog is gradually lost due to the now lysosomes. Fertilization: the acrosome of spermatozoa contain enzyme which help in penetration of white line membrane of egg during fertilization. Phagocytosis: lysosome digests the leukocyte, foreign proteins, bacteria and viruses. Cell division: lysosomes are helpful to initiate mitosis and meiosis in the cell. Structure of, now see the structure of lysosome. Now within the structure of lysosome, now the granules are present and it has outer membrane and inner membrane. Outer membrane is smooth, inner membrane now which is consist of granules. Then the next, another cell organelle, now that is the ribosomes. These are ultra microscopic granular particles which occur in both prokaryotic and eukaryotic cells. They may be present freely in the cytoplasm or attached to the endoplasmic reticulum, mitochondria, chloroplast, and nucleus. Ribosomes were first discovered by Paladin in 1955 in eukaryotic cells. These are involved in the protein synthesis process. For example, pancreatic cells, then the mammary glands, now cells. Types of ribosomes according to the size and sedimentation coefficient value. These are classified into now two groups. Now the first, 17S ribosomes. These are relatively smaller in size and have 17S sedimentation coefficient value. These ribosomes occur in prokaryotic now the cells, mitochondria and chloroplast. Then the second, 18S ribosome. These are comparatively bigger in size and have 18S coefficient now value. These are occur in eukaryotic cells of plants and animals. Now see within the types of ribosomes within the now diagram. Now 13s and 15s, 17s ribosomes, 18s ribosome. It also consists of 2 units, 14s and now these 18s subunits. This is regarding the types of now the ribosomes. Structure of now the ribosomes. These are small vesicle-like structure. It may be oval or it may be globular in shape. Each ribosome is porous and is composed of now 2 subunits. The bigger unit it is large and it is dome shaped, while the smaller subunit it is small and it is now cap like. These two ribosomal subunits remain associated with each other in low concentration of magnesium ion. But when magnesium ion absent, they remain separated. Sometime two ribosomal become associated with one another, form a dimer. During protein synthesis, many ribosomal get now aggregated on a messenger RNA to form polyribosome, or it is also known as polysomes. Now we see within the diagram structure of now ribosome. It consists of now these two subunits, and after combination of these two subunits, one unit which is smaller in size, another unit which is bigger in size, then it combine and then it forms now the dimer. Then after within protein synthesis, polyribosome or polysomes are formed. Now see the subunits 14s, then now these 16s in combination with now magnesium ions, then it forms now the ribosomes. Like that, so now many structures are formed, and this structure it is generally known as polysomes. Functions of ribosomes. Functions of ribosomes. The ribosome is complex molecular machine found within all living cells that serve as the site of biological protein synthesis. Ribosomes link amino acid together in order to specified by messenger RNA molecules. Ribosome act as a catalyst in two extremely important biological processes called the peptidyl transfer or peptidyl hydrolysis process. Thank you.